Good morning to everybody, and we are ready for this fourth webinar of the Eufresco Basics. Eufresco Basic is a European project that aims to find alternative to synthetic pesticides, and among those there are the basic substances that are compounds that are already used in human medicine or as a food, and we can use them or try to see if they works toward the pest and pathogen of plants. So since we already eat those compounds, they does not need to be proved for their effects on humans, animals, and the environment. Then registration is easier, it is longer, there are no LMR and so on. There is a already approved list of 23 basic substances, but several others are candidates. So today we will have a couple of very interesting presentations from Simona Sanzani and from Luis Gonzalez Candelas. I take you the greetings from Baldisera Giovanni that had at the same time another meeting, so he could not skip. So he say hello to all of you. Baldisera Giovanni is a Eufresco coordinator and he supported us a lot during this project. So making it short, I will give the floor to Simona Sanzani, that she's a researcher at CEAM in Bari. She will have a presentation titled The Basic Substance and the Candidate 1 as a Resistance Inducer to Fungal Disease. Simona, you are welcome to show your presentation.
Thank you, and good afternoon to everybody. It has been estimated that up to 45% of fruits and vegetables are lost along the production chain from the cultivation till the consumption. And the relevance of these losses at the different stages varies according to the level of industrialization of each single country's geographic area. But it includes all the different steps of the chain. So that recently the European Commission emitted a document called the Green Deal, in which stressed about the importance of a sustainable approach to food and feed losses that takes into consideration each single step, including the consumption. As in the industrialized country, most of the losses take place at the consumer's house. Most of the losses, or at least a huge amount, can be associated to biotic stresses, and the public, we are used to think that the most important way to counteract them is the use of antimicrobials. But we should keep into account that we have another ally, which is the host plant or the fruit, if I'm a hypothalamus pathologist, that can help us in counteract the pathogen, and we can exploit its natural defense mechanism, which could be somehow alerted so that they can be more effective in controlling the pathogen. And this is basically the topic of my speech today, associated to the basic substances, which is at the center of this, which are the center of this project. Apart from protecting, reducing the losses, those treatment that induce resistance increase also the content of compounds which have a significant not only from a disease control perspective, but also for the positive effect on human health. I'm thinking to the most famous resveratrol, but there are many others, as quercetin, which is an antioxidant, anti-inflammatory molecule. More recently, the phenolic compounds contained in the pomegranate juices, so we can end up with an added value product. So what I would like to do today is to give some examples of the use of resistance inducers, which are in the basic substance category, or maybe in the future could enter into this category, applied to the different steps of the production chain, starting from the cultivation. In this case, I want to make the examples of the protein hydrolysates. Protein hydrolysates are a mixture of amino acids, which can be of vegetable or animal origin, which are obtained by a chemical or enzymatic process. These amino acids, apart from having some direct effect, I mean their own effect, for example, promoting growth, their synthesis is very expensive from an energetic point of view for the plant. So if we give them exogenously to the plant, it can save energy that can be devoted to other biological processes, including defensive mechanisms. We tested, in particular, several protein hydrolysates, mainly from vegetable origins, but also one from casein, which are already sold as dietary supplement or used in the food industry. In particular, we tested them against otitis cinerea, a relevant pathogen for many commodities. In this case, we were more interested into grapes. And we noticed that none of these treatments were able to control the growth of the pathogen in vitro tests. However, when we tested them on berries, on harvested berries, they were able, especially the soybean and the casein hydrolysates, were able to control the disease incidence and the disease severity. What's more, we tested them also on a bigger semi-commercial scale. We conducted some assays in greenhouses and in the field against downy mildew and grey mold, which are two relevant diseases of grapes, in particular table grapes in this case. And we noticed that the two treatments, in particular the soybean one, was effective in counteracting the disease severity. So, considering that in previous assay they weren't effective on the pathogen itself, at least on otitis cinerea, we hypothesized that they may act by inducing resistance. That's why we conducted some specific tests. For example, we evaluated the content of cytoplasmic calcium. I mean the changes, the increase and reduction in the content of cytoplasmic calcium, because calcium is a powerful secondary messenger of several metabolic functions, including defense responses. And in fact, we noticed that as compared to oligolacturonide as a positive control, let's say, they were able to, both soybean and casein were able to cause an increase in concentration of cytoplasmic calcium. That particularly for casein, this was a dose effect, whereas in the case of soybean, the two higher concentration basically had the same effect. Interestingly, the soybean peaked at 2 minutes, while the effect of casein was a little bit slower, 5 minutes. Then we tested the production of MAP kinases, which are also proteins involved in several cell functions, including differentiation, growth, differentiation, and that. And we noticed that both in presence of soybean and casein, there was a peak in the production of these proteins at in the concentration of this protein at 15-30 minutes from treatment application. Then there was a decline that, however, in the case of soybean was much less evident than as far as casein concerned. As a third step, we evaluated the differential expression of genes known to be involved in defensive responses. As you can see, we have several PR proteins, plus other genes as the stilbene synthase, which is involved in the synthesis of resveratrol, as mentioned before. These are it is represented a differential expression as compared to the antithetic control. And as you can see from the graphs, both casein, but above all soybean, cause an induction in the expression of these genes. This induction peaked at within 24 hours after treatment application. And it is particularly interesting as a response which is prompt is more probably that might be successful. But interestingly, in some cases, the peak arrived later after 24 hours. And this is also a positive circumstance in our opinion, since it might guarantee a more long-lasting protection. Finally, we quantify the production of the end products, let's say, of these defensive reactions, pathways, which is the phytolexins, so resveratrol, and its derivative, delta-viniferin and epsilon-viniferin. As you can see here from the graphs, both in presence of the two treatment, there was a higher production as compared to the control, particularly in presence of the soybean protein hydrolysate. The production was again quite took place quite early in the range 8 24 hours, 36 hours also. And this again seemed to support the hypothesis that protein hydrolysates, particularly the soybean one, could work by inducing resistance in the host. Concerning instead the post-harvest phase, I would like to make an example of the phenolic compound quercetin, which is present as a for someone as a phytolexins, for some other as a phytoanticipin. There is a debate on this. And as antioxidant and anti-inflammatory properties, so that you can buy it as a dietary supplement. What we did is to test them against blue mold, which by Penicillium expansum, which affects mainly palm fruit, but many other commodities. Penicillium expansum also produces a mycotoxin, which is called patulin, which is regulated by the European Commission because it's considered detrimental for human health. As you see, we tested quercetin first in vitro. It wasn't able to control the growth of the fungus, but prevented the production, reduced the production of patulin by 40% roughly. What's more, when we tested in vivo, the activity was much better than we expected. It reached up 90%, and the patulin was reduced by 70%. So this good effect observed in vivo, which do not correspond to a direct effect on the fungus, let us hypothesize my work by using resistance. And in fact, we tested the genes which were differentially expressed because treatment application following quercetin application in apple. We did it by a technique which was called suppression subtractive hybridization. And we end up with a series of genes coding for protein, which were included in different functional categories. I would mention the most relevant according to me, which are metabolism, protein with binding or cofactor requirement, and cell rescue defense and virulence, which includes the response to oxidative, protein involving the response to oxidative stress, and this is in line with the quercetin antioxidant properties. The most represented protein were those associated with the production of one protein, which are involved in the degradation of mRNA present because of pathogen attack, with also a good representation of the so-called phenenzyme biosynthesis protein, which counteracts the oxidative stress caused by microbial attack, and the 26S proteasome regulatory particles involved in the degradation of damaged or annealed protein. These results were confirmed by a traditional approach, which is real-time PCR evaluation of differential expression of the genes. And the results were confirmed, and some genes were highlighted for being particularly induced. So we decided to further test this 4 gene plus the 26 regulatory particles that I mentioned before in an experiment, in a time course experiment, which was meant to assess the effect of the wounding as a stressing event, as quercetin application, as a standalone without taking into consideration the wounding, and the combination of the two stresses, let's say. What we noticed was that the 5 genes selected were expressed even following wounding. So they were all transcribed even in quercetin untreated fruits. But in presence of quercetin, considering the quercetin application as standalone, this expression was higher and in many instances anticipated. And as said before, a quick response is more probable to be effective. Finally, and interestingly, in our opinion, summing the factor of the two treatment, the two stresses, we observe a even much higher induction of these genes, which apart from being higher was also more long-lasting. And as already said, this is also a positive circumstance for allowing a certain level of protection. So all these clues went towards the idea of quercetin as a resistance inducer. What is also surprising and interesting, in my opinion, is the fact that even a sanitizer treatment, as we consider salts, sanitizers, we think to sanitizers as something that acts directly on the pathogen, and so we are not used to associate them with the induction of resistance. However, they are very common, since most of them are included in the category of the generally recognized as safe by the American FDA, or as the case of sodium bicarbonate are already in the category of basic substances. As confirmation of their direct effect, we test them against Penicillium digitatum on citrus, causal agent of green mold. We tested several salts, and surprisingly, 2 of the sodium carbonate and bicarbonate and potassium carbonate and bicarbonate were very highly effective at 3% dose, taking into account that we were treating fruits that have a tough peel, were effective in completely preventing the disease. And this is somehow, I won't say expected, but reasonable. But what surprised us was that when we tested them in 2 close but different wounds from the pathogen, I mean that the pathogen and the salt didn't came into direct contact, although the effect was much lower than that observed previously, it was anyway significant and satisfactory. So even in this case, we try to understand something more about their mode of action. And so we decided to test the activity of enzymes known to be involved in defense mechanism. I will start with the most famous PAL, phenylalanine ammonium, which is involved in the shikimate pathway, which leads to the production of phenolic compounds and many other defensive mechanisms, including also chitinase, glucanase, and peroxidase involved in the response of those to the stress and to pathogen attack. And we noticed that sodium carbonate and bicarbonate cause an increase in activity of particularly of PAL, but also of glucanase and peroxidase. And this was a first clue toward the reduction of resistance. Then, even in this case, we decided to quantify the phytolexins present in the peel of the fruit following the treatment. And we observed that, in particular, we tested umbelliferone, scopoletin, and scoparon, and we noticed that the 3-phytolexin increased because of treatment application. Particularly, there was a peak for the umbelliferone within the 24 hours. At 12 hours for scopoletin, much later and much strongly for scoparon at 72 hours, thus supporting also the consideration that since scopoletin and umbelliferone scopoletin after the peak declined, and this decline happened sooner than the peak in the content of scoparon, umbelliferone scopoletin might be precursor of scoparon. We tested also the production of the content of sugars, in particular glucose, fructose, and sucrose, and noticed that because of sodium bicarbonate application, there was an increase in the production of the content of sucrose, and consequently a decrease in glucose and fructose. And this is interesting since sucrose is the precursor of phenylalanine, and which, as I said before, is the starting point of many biological processes that leads also to defense mechanisms. So even in this case, we have some proof that seems to support the idea of resistance, the induction of resistance. Interestingly, this resistance inducer seem also to function as resistance inducer also if applied in as electrolyte in electrolysis reaction. Professor Ippolito told us about the use of electrolyzed water as a defense, as a sanitizer treatment. For example, it was effective against Penicillium digitatum and the washing water of citrus, and against rots on the fruits which were moimented in that water. But today I want to make a step further and report about some results concerning the use of electrolyzed sodium bicarbonate as a resistance inducer. I want to make a clarification that the concentration in this case is roughly half of that was used in the previous essay I showed. Anyway, when the electrolyzed sodium bicarbonate was applied in a close but different fruits from that of Penicillium digitatum, it was anyhow effective, in some instances better than its not electrolyzed counterpart, and induce the activity of the defensive enzymes we saw already before, with good results, particularly on PAL and peroxidase, but also on chitinase. In the case of the glucanase, the activity was lower than that of its not electrolytes counterpart, but it took the peak took place sooner, so maybe this could contribute to its effectivity. So, arriving to the conclusion, I want to, the sense of this speech today was to draw the attention on the fact that an effective level of control can be obtained even with the aid of resistance inducer, and that induction of resistance has several positive features. For example, apart from as I mentioned, the chance to have a product which contains higher level of beneficial, I would say, nutraceutical products, reasonably being the induction resistance a complex mechanism, it could be more difficultly overcome by pathogens compared to antimicrobials. So I would like to thank all the people that contributed to the researches and you for your attention.
Thank you, Simona, for the very interesting presentation, and the floor is open for questions, and anybody that wants to have questions can raise the hands or open the microphone and ask the question. So since the first will come, Luis.
I was afraid of Luis.
It's a very friendly, just it's not a question, it's just a matter of curiosity. I was wondering, the protein hydrolysis you are working with for induction of resistance are commercial products, right?
Yeah.
So you don't know exactly how variability is if you conduct the hydrolysis by yourself compared to a commercial product coming from a particular company.
Yeah, it's true. Yes, I guess that the next step, which I would like to do, is also to assess the precise composition, because on the label there is a certain indication. I guess that it might change also according to the batch, and so it would be nice to know.
Soybean is also a basic substance, right?
No, I guess not. No.
Okay, so
maybe we can support the application.
Yeah, the application. That's right.
No,
that was all.
If I can add something, the hydrolysis is for sure enzymatic by enzymes, not chemical.
Okay.
Because I remember I visited the industry. They had a big container in which they processed the different matrices, and was enzymatic. And the enzymatic hydrolysis is quite different than chemical, because you have a shortened, shortened amino acids or peptide, and that are more effective in case of application to plants.
Yes, if I remember well, I don't know how to say in English, but in Latin is levogiro,
yes,
L and D. So I guess the L is more easily absorbed by the plant as compared to the D, if I remember well. So that's why there's a difference between the enzymatic and chemical, because the enzymatic produces a higher quantity of amino acids which are available from the plant.
Very good. There are other questions for Simona. I have one from me. Simona, your data told a very interesting story, and from different studies that you mentioned, you can have a different trend. According to you, how long it can last, these induced resistance? Of course, it depends on the system, but which is your opinion about?
So we conducted mainly studies at lab scale, let's say, although we had also an experience with for apples commercial conditions. And we assayed the fruits for 1 month, and until 1 month there was still a good containment of the disease. So I can testify for this, and but it would be nice to conduct larger scale trials, maybe in companies or in storage packing houses will be interested.
Thank you. Additional questions? Okay, you can save them for the general discussion at the end, because now we can give the floor to Luis Gonzalez Candelas with a talk titled "Can Calends Become Part of the Toolbox to Control Post-Harvest Disease of Fruit?" Luis, the floor is yours.
Okay, thank you very much, Gianfranco. Do you see my screen? Do you see my screen, I guess? Okay.
Yes, we can see.
Okay. So I'm gonna try to present the work we are involved during last years, and not only that, but trying to explain how we arrived to this point, coming from different kind of approaches to post-harvest pathology. I don't know what's going on.
This was last slide.
Okay. I need to go to the very beginning. Okay. So here are the pathosystems we are working during last years in our group in Valencia. Most of you know that we are in a region where citrus are grown, and the major pathogens of citrus fruits are Penicillium digitatum and Penicillium italicum. But we are also working on 2 different pathogens that are producers of mycotoxins, such as Penicillium expansum, producer of patulin or citrullin, and also Aspergillus carbonarius or Clatoxin A. Most of our work has been dedicated to basic aspects of pathogenesis and resistance on the fruits. And we try to explain how, from these studies, we end up with basic substances, because I guess many of you will wonder, what is this guy doing here, working with applied aspects? So some years ago, we conducted a study to study the genes that are induced during the infection of citrus fruits by Penicillium digitatum. We conducted the studies from both sides. On one side, we studied the citrus genes that were upregulated by induction, and on the other side, the genes, the fungal genes that were induced during infection. Okay, this is the same kind of study that Simona presented you before, SSH, suppression substrate hybridization. At the end of the process, I'm not going to go into details, we end up with a cDNA library enriched in induced genes, and we conducted both hybridization and sequencing of the clones. And one of the things that caught us the attention was the high number of genes encoding proteases that showed up on top of the list. Here you see labeled on pinky color, different proteases just on top: aspartic endopeptidase, aspergillopepsidin, tripeptidyls, carboxypeptidase. We also found many genes coding for cell wall degrading enzymes, those labeled in blue. But we were wondering, what are these proteases doing during the infection? So as there are so many different proteases, here you can see a more detailed study of the expression of these genes during the infection process. Compared to the in vitro grown, you can see that most of them are highly induced just during infection. We confirmed these studies later on by another approach, that is a RNA-seq transcriptomic approach. But the problem with having so many genes coding for proteins that are similar is that if you knock out a single gene, another gene may take over the one you remove. So we decided to go after a regulatory protein of proteases. A few years before, the gene called PRTT was described in Aspergillus, both in Niger and Fumigatus, as a regulator of proteases. So we bet, okay, maybe this regulator, if it does exist in Penicillium digitatum, it may control the proteases that we are seeing here during infection. And the hypothesis was, okay, if we remove the regulatory gene, we may decrease drastically the amount of proteases during infection, and we will see an effect. Okay, so we started the work, we constructed the knockout mutants, and finally, after physiological and phenotypical characterization, we conducted the gold assays, infection assays. This is one experiment. We did 3 or 4 different experiments, and in all cases was the same. The knockout mutants, those labeled with a triangle, didn't show any effect on pathogenicity. So we wonder, we didn't expect that, because during in vitro assays, we clearly see a reduction of proteolytic halos around the colonies in the mutants. So we analyzed the expression of the 2 major proteases that we got in the previous study. And we observed that in the deletant, those proteases were still being produced, at least the genes were still being expressed. So we gave up this approach, but we were still interested in knowing what the proteases were doing in this process, during the infection process. So we took another approach, a pharmacological approach. So we applied a cocktail of protease inhibitors, and this is the first results we obtained. Wow, this looked amazing. Just by adding a cocktail of proteases, the effect on infection was huge. Using 2 different amounts of cocktail, the reduction in one case was 100%. So we wonder, well, what is in these cocktails that is capable of doing this high suppression of infectivity in Penicillium digitatum? As the cocktail is a commercial product, we know what is in the cocktail. The cocktail is made of these 4 different proteases: ABCF, E64, pepstatin A, and 1,10-phenanthroline. Each compound inhibits specifically one family of proteases. So we make the breakdown of the cocktail using single compounds, combination of 2 or 3, and you can see here the results of one of those experiments. Whenever phenanthroline is present, there is no infection. Okay, phenanthroline in the cocktail, when we use the cocktail of 0.1%, the equivalent amount of phenanthroline is 0.5 millimolar, and in the case of 1% cocktail, it's 5 millimolar, and in our assay as a pure compound, we use 1 millimolar. And you can see here the dramatical effect of phenanthroline. So phenanthroline is really responsible of the effect of as a protease inhibitor. Phenanthroline is an inhibitor of metalloproteases. So we wonder, other metalloproteases are also able to do the same? We conducted the same kind of assays using another set of metalloprotease inhibitors, such as bestatin or phosphoramidone on one side, and on the other side, we use chelants, because phenanthroline is a metalloprotease inhibitor, but it's a chelant. It's a strong chelant that is commercially used to detect iron 3. The union between phenanthroline and iron 3 gives a red color that is easily measurable in the spectrophotometer. So we included different chelants such as EDTA, EGTA, ferrocin, and DETC. Well, the first results we observed is that the metalloprotease inhibitors, at least the ones we use, they didn't do anything. You can see the control, the progression curve. compared to the metalloprotease inhibitors. The phenanthrone is run here. There is nothing. The chelans, well, they did something, not as much as the phenanthrone, but EDTA and ferrocin, they reduce the infection degree. So maybe the effect of phenanthrone is not general as a metalloprotease inhibitor, because other inhibitors that depend on another mechanism of action don't protect the infection. However, some chelans are able to reduce, not as strong, but they're able to reduce the infectivity of Penicillium digitatum. We also tested the effect of phenanthrone on other strains and on other species of penicillium. Here are the assays of Penicillium digitatum, this strain, and this is resistant to fungicide, and this is susceptible to imazaline. So the effect of phenanthrone is very similar to the effect we observe with imazaline, and it's also effective with Penicillium italicum and with Penicillium expansum, although in this case we had to increase the concentration of phenanthrone. But here you see at day 3 we have almost 100% infection in apples and nothing when we apply phenanthrone. So we were really excited about this compound. This compound is a chemical compound that is classic, is very cheap. So this is the aspect of the apple fruits. After, I guess I didn't write it, I guess it's 1 week after infection at 20 degrees. So it's dramatic, the effect. We started working on the possibilities of phenanthrone as a potential control agent. So we wonder whether it was able to protect the infection when applied as a curative treatment. That is, right 24 hours after the pathogen was inoculated. Here you have an assay in which the fruits were inoculated with Penicillium digitatum in wounds, and 1 day after that, the fruits were dipped in phenanthrone and then were stored for 4, 5 to 7 days at 20 degrees. Here is the evolution of the infected wounds with phenanthrone. Okay, you see the clear reduction. But if you see the picture here down at the bottom, you can see that there is a kind of pitting here at 10 millimolar. 10 millimolar is one excess. If you look closer, you see some pitting. So we were worried about the possible toxic effect of an excess amount of phenanthrone. Here we observe a halo around the wound because we use a kind of rubber stopper to make the wounds, and we make wound after wound. So we leave a mark of the rubber stopper surrounding the wound, and this is the mark that you can see here. It's also observed here. This is when we inoculate the phenanthrone just into the wound with a pipette, or here when you immerse the fruit in the solution containing phenanthrone. Anyway, in another assay, here is also checking for curative effect and cold storage. So the fruits were inoculated. 1 day later, were immersed in phenanthrone, and they were stored for 4 weeks at 2 Celsius degrees and 1 week at 20. The effect of protective effect is quite good. And in this case, the phytotoxic effect was not so clear, even at 10 millimolar. Here you have a zoom out of the oranges. So we observe this phytotoxicity, some instances with oranges, but we haven't observed this effect on apples. Here you have also a curative treatment of phenanthrone with on apples. The apples were inoculated with Penicillium expansum. 18 hours later, they were immersed with phenanthrone, and fruits were stored 4 weeks at 2 degrees, and then 1 week at 20 degrees and 90% humidity. And here you see the aspect of the apples after the immersion with phenanthrone. There is no clear phytotoxic effect, and these are this is the picture of the fruits after 1 week 4 weeks at 2 degrees and 7 days at 20. So how is the phenanthrene working? Is just inhibiting a metalloprotease? We don't think so. As a matter of fact, when we conduct antimicrobial, antifungal assays using microtiter plates, we observe that phenanthrene clearly is able to inhibit the growth of Penicillium digitatum, Penicillium italicum, or Penicillium expansum with MIC values ranging between 25 and 50 micromolar. Here, there is no inhibition of protease as being a limiting factor for anything, because in this case, you are growing the fungus in an in vitro culture medium. So there must be something else that is important for the growth of the fungus. We also check whether the effect of phenanthrene was either fungicide or fungistatic. So when we apply the MIC concentration to the spores and we leave the compound to be in contact with the spores for up to 2 days, we observe a reduction in viability of the spores, but a reduction that does not reach even 50%. So this does explain a fungicidal action of phenanthrene. We think that most of the major action is fungistatic. Moreover, if we include some metal ions into our assays, we are able to revert the inhibition shown by phenanthrene. Here, if you focus on the left upper graph, you will see a typical experiment adding 0.8 micromolar. This is 13 micromolar or 20 micromolar phenanthrene. As you increase the amount of phenanthrene, the fungus cannot grow. But when you supply 2 micromolar of either iron as iron sulfate or zinc sulfate or copper sulfate, you see that at the concentrations that it was not able to grow, now the fungus is able to grow. And the best ion to reverse the inhibition of the phenanthrene is iron. You see here that when we add iron, the fungus is able to grow at 20 micromolar phenanthrene. When it's zinc or copper, those 2 ions cannot reverse the inhibition. So we end up showing the effect of phenanthrene is basically because it's able to chelate some metal ions, because when you add metal ions, you reverse this effect. But this is not only during in vitro growth. When we add the same kind, when we carry out the same kind of experiment in vivo, we observe the same. Just focus on this left part of the graph. This is the evolution of the infection when Penicillium digitatum is infected and when phenanthrene was added. If we added, besides phenanthrene, zinc or copper or manganese of iron 2, this is the result. With zinc and iron, you recover completely the infection of Penicillium digitatum. So it means that during the in vivo assays is the chelation of zinc and iron 2, at least from this point of view, the ions that seems to be important. As a matter of fact, when you add those ions alone with Penicillium digitatum, the right part of the graph, you see that the evolution of the infection is faster. Here you can see what is the evolution with zinc and with iron 2 when compared with no added metal ion. So this is telling us that, or suggesting that during the infection, the fungus is facing a limiting amount of several micronutrients. And this wonders, okay, this is until now is the effect of phenanthrene. This is a chelan compound. Are other chelans able to do something similar as we conducted during the very first assays when we compare metal proteinase inhibitors and chelans to see And we saw that chelants, they did something not as good as phenanthrone. So we wonder, EDTA could do the same or something similar? Well, we first started checking in vitro. In vitro, this is a growth in GPI, in microtiter plates, showing the effect of different amounts of either phenanthrone or EDTA. And here you have the MIC values. Okay, in vitro, EDTA is worse, but it's not much worse than phenanthrone. The problem seems to start with the very nature of the action. A chelant is able to sequester the metal ion. And so depending on the composition of the medium, you will have different amount of different ions. So when you compare the same amount of EDTA in two different assays using two different medium, GPI is glucose, peptone, these extracts, and PDB, you see that the difference in activity could be very high. From a MIC of 313 in this particular assays, we are conducting the assay much longer than in the upper part of the slide. You move to more than 2,000. This is 2 millimolar, 2.5 millimolar. So this is telling us that the effect of the chelant is going to be dependent, among other factors, on the composition of the medium. So what we are now working on is we have modified Penicillium digitatum introducing GFP. GFP is a fluorescent protein, as you can see here on this part of the microtiter plates. This microtiter plate is full of orange disc. Just we take with a cork bottle the ring, and we put the ring upside down inside each of these microwell plates, and we inoculate the different wells with the fluorescence strain. So we can follow the fluorescence as a signal of the growth of the fungus. Okay. As a matter of fact, this plate that I'm showing correspond to no EDTA or different amounts of EDTA added at the same time with the ring disc. And we can follow in a spectrophotometer, in a plate reader, the fluorescence of the different wells. So we can end up with curves that look like a classical microbiological growth curve, where you have the no EDTA added or EDTA at different concentrations. Okay. So when we conduct these assays, this is what we're doing right now. So those are results from last week. When we are conducting these assays with discs that have been inoculated with Penicillium digitatum, and this is Penicillium digitatum, this is also Penicillium digitatum, and all these are EDTA alone here and also here, and the rest are EDTA supplemented with calcium, magnesium, manganesium, copper, zinc, iron 2, iron 3, or cobalt. As you can see here, only iron 2 or iron 3 are able to partially revert the inhibition of EDTA. So this is how we end up from a completely basic study of the mechanism of pathogenicity to a potential basic substances, because as most of you know, EDTA is additive that is widely used in the food industry, and we think that it has a potential to be used as an antifungal. It's not going to be as effective as a commercial fungicide, but it's able to, at certain degrees, to exert some control. Those assays that I'm showing you here are conducted with 20 millimolar. 20 millimolar EDTA is 0.74% EDTA. It's a reasonable amount. I mean, with chitosan, you usually work in the range of 0.4, 0.5, 1%. So it's in the same range of amount. And that's why we wanted to join you in this Fresco meeting. As a matter of fact, the use of chelants is something that at least I haven't heard before in the field of post-harvest pathology. That is my field, okay? But if we go outside our field, we can see that this is not new at all. As a matter of fact, there is a concept that was coined many years ago that is nutritional immunity. That is something that we have, all mammals. One of the mechanisms that mammals have in our immunity system to fight against pathogens is to remove metal ions from the bloodstream in order to avoid pathogens to grow. We try to, there are different ways to do that. You can sequester a chelant inside the cells. We produce more transferrin, that is a iron binding protein that is produced in response to our one inflammatory processes. So in immunology, this system of starving the pathogens by removing metals from the medium is already known and is well documented. As is the case, here are the it's a sketch of different fungal pathogens, human fungal pathogens, Aspergillus fumigatus, Rhizopus, or Candida albicans, how they handle the battle for different ions compared to the mammal system. So as a conclusion, we can say that there are some chelants of transition metal ions that can be used as antifungal agents with curative action to control the development of infection caused by penicillium in citrus and apple fruits. And we expect, or we want to see whether some additives can be fitted within this frame. And the study of the pathogen's virulence mechanisms can show us new opportunities for disease control that are worth to be investigated. This is my take-home message, and that was all. Yes. Thank you for attention. I'm open for your questions.
Thank you, Luis, for the very interesting presentation. I'm sure that a lot of questions will come from the audience. I was surprised from your talk about knowing that EDTA and other chelants are used in food industry. Do you have further detail about this?
Yeah, EDTA, yeah, it's a very common additive. It's, I don't remember the name. I think it's E585, and it's widely used as an antioxidant. But antioxidants, some antioxidants are good antioxidants because they are able to remove ions. And you see, iron 2, iron 3 is one of the couples of ions that are more responsible of oxidations in food.
Excellent. I'm sure that other questions will come from the audience. Who wants to start? Simona.
No, just a curiosity. As far as understood, there is a difference in the susceptibility of the different species of penicillium. It would be nice to test even other genera and see if there are differences, stronger differences, or the reason why.
I don't know how to tell you. The facts are the numbers. Yes. All the fungi that we have tested are susceptible, but they are not always susceptible as Penicillium digitatum. Penicillium digitatum is among the most susceptible fungi that we have analyzed. But botrytis is also susceptible, Rhizopus also, Alternaria is also susceptible. But we don't know why some of them are more susceptible to others. We speculate that it depends, the effect we observe, this is hypothesis, okay? We think can be explained in two ways that are complementary, they are not exclusive. One is that you remove the metal ions from the medium, okay? And if you only do that, the fungus is still able to grow with their own store because the spore has some metal inside, and even if you deprive the ion from outside, it's able to keep going on for a while with internal metal that contains And we know that the best chelants are those that are able to get into the cell. So EDTA is not as good as phenanthrolene, we think, is because phenanthrolene is able to permeate the membranes, whereas EDTA is not.
Yes, in fact, I did some essays in which we observed a lower effect than expected because the compounds weren't able to penetrate into the conidia. So, but I guess the chitosan is a good carrier. I don't know, Gianfranco.
Yeah, it is.
Well, things.
I mean, the final idea would be if EDTA is not very good, maybe in combination with some other additive that is not antimicrobial or has very poor antimicrobial activity, maybe there is a synergies that has to be undisclosed yet in order to improve the antimicrobial activity. This is what we are going after.
Thank you.
I don't know whether we will succeed or not, but I think it's worth to give a try.
The next differential expression experiment.
Yeah, it was one.
Very good. Additional question to Luis or to Simona? Who want to start?
You have an open hand. Sarah Mojela, raise the hand. Gianfranco.
Yes, I don't see this.
A raised hand over there. Sarah,
are there?
I see the hand.
Okay. I don't know. I saw Professor Ruggero Osler in the audience. He is an expert of induced resistance, although from other fields. I don't know if, Ruggero, you have any question. I know that the subject is a little bit far as compared to your experience, but in case you want to ask something to the speakers, you are more than welcome.
Thank you, my friend. I have to confess that this is not my personal field that I know the best. I must to say that I am a plant pathologist of the open field and not of the laboratory. Anyway, I would like to put the same question you did before. To me, one of the most interesting things to speak about these inducers is how long is the persistence of their input in the plant. And secondly, I understand that we want to understand if the effect is direct on the pathogen or indirect on the plant. To me, in the nature, the most common probably is the dual effect. We don't have so nervous to pretend to know if whether one or the other. Probably are both more one or less the other, but both. This is my opinion, of course. I thank you very much for your question. Thank you.
Who want to start? Luis and later Simona. Although I have not presented anything related to induced resistance, Simona knows I've been working also with induced resistance, and I agree with your opinion. In many instances, you cannot see the effect is antimicrobial effect alone or induced resistance alone. In most cases, you have partial effect as antimicrobial and partial effect as as inducer of resistance. So sweeter the better, we say here in Spain. So you have two effects in one compound, better.
Yes,
if it is three, even more.
Yeah, sure.
It's trial.
I want to add also that, I also conducted some studies on the role of secondary metabolites. So maybe there is a third option in which the treatment does not affect the primary metabolism of the fungus, but the secondary one. And as we all know, many secondary metabolites have a significance in the disease onset. I'm thinking of phytotoxins, but now also there are some studies in which I was involved regarding the role in pathogenicity of mycotoxins. So I totally agree with Professor Osler, and I would add also a third option from my side.
Excellent. I saw in the middle of the screen Alessandra that is coordinating a special issue of the horticultural journal, basic on alternative to synthetic fungicides. Alessandra, do you have any question for the speakers?
Yeah, I have a curiosity for Luis about the use of this kind of chelators, such as a DTA, can compromise the food safety or quality, or maybe the possibility to have some particular residue in food?
I don't know yet. So we are in the previous phase. The only thing I can tell you is that the EDTA is approved by European Food Safety Agency as an additive, as has a very long story of being used in the food industry. So from that point of view, in general, should not be of great concern. But what is the effect, a specific effect of EDTA on fruit? We haven't conducted those assays yet. We are conducting the assays I show you with discs, because we are just starting to get into the harvesting season. So we will start conducting fruit assays with whole fruit in November, next month. So what we are doing now is buying the fruits in the supermarket that are treated with fungicides. So that's why we remove the ring and put upside down to conduct the assays, because the fungicides get very small, they cover the surface and get into the peel, but just few millimeters. So by taking the disc and inoculating the white part, we are far away from the fungicide. So until we don't have fresh fruit, we cannot conduct the assays with EDTA. Just by immersing the fruit. I don't expect, but I mean, you never know. I don't
as this kind of chelator, but in the field, in the environment, can cause great problems.
It could be.
Yeah, in the soil, maybe.
Yeah, in the soil. Yeah.
So for this reason, I thought that maybe by using these on fruits or food can cause some kind.
Depends on where do you use it, because for example, now here in the packing houses, at least in Spain, the fungicides, the way the fungicides are applied are either in water, and the fruit is either drenched or immersed, and the residue, the water that is residual, cannot be thrown to the pipe, has to be dispensed specifically. In these cases, there is no disposal of whatever chemical into the environment. So from that point of view, another thing would be if you want to apply this on the field. If you want to apply it on the field, would you irrigate? That is a completely different story. But to be applied in a contained system that is a packing house, I mean, I'm not an expert on that area, but the compound should not get outside the packing house in the drain system. That for sure.
Okay, thank you very much.
Welcome.
Very good discussion. Other comments from the audience to the speakers? Of course, you can be free to contact them and to ask further details. And the ones of you that maybe reached later or want to see again the presentation, can find them on the Facebook page, and I say hello to the people that are joining, are following us from Facebook, or the video at high resolution will be on the YouTube channel. And thanks to Lucrezia that is recording and will post it soon. If there are, I don't see other hands raised, so I thank the speakers for the excellent talk, and you were very, very kind. And I'm pretty sure that we will see again the speakers, for the ones that are interested in the field, in the post-harvest pathology congress that will be in May, June of the next year. And the ones that are interested in the field know that, or maybe aware that the deadline for abstract submission is the end of November. So if you want to show something, you can run fast, write down your results, and present to the organizer, because they are placing together an excellent program. So I thank all of you, and for today, I think it's okay. We were highly satisfied by the presentation. We had a very good sharing of knowledge. And the next appointment, you can mark the calendar, is on November 23, where the first speaker will be Jolanta Kowalska, that we can see here in the middle of the screen. She will talk us about basic substance and the potential basic substance for Phytophthora infestans and for seed-borne pathogens. So we will have Clemencia Chavez Lopez that will talk about root essential oil, and Elizabeth Caraffa that she's in the audience as well, and she will talk about pre and post-harvest chitosan application for sour cherry. So a lot of other information are ready for us for November 23, so save the date and see you at that time. Enjoy your evening. Hello to everybody.